Shri Shivaji Vidya Mandir, Aund. Welcome to our biology class. Today we are going to see that circulatory system in human. In a previous lecture we have completed the respiratory system. Now we will continue with the circulatory system in the human. Basically, human circulatory system is made up of a blood vascular system and the lymphatic system. Lymphatic system means blood cells. So first we will see the blood vascular system and after that we will see the lymphatic system. First of all, this blood vascular system consists of heart as well as blood vessels along with blood, and there are blood composition. So this blood is made up of a plasma and the blood cells. Basically, in the blood, plasma is present about 55% and in the blood cells are present about 44%. And this plasma is basically fluid and blood cells are the cells. This plasma and blood cells can be separated by the centrifugation method, and by the centrifugation method this plasma and blood cells can be get separate out. Study of blood is called as a hematology. Why it is called as a hematology? Because in the blood there is a hemoglobin is present, and on the basis of word hemoglobin this hematology word derived. We know blood has a red color, and in the blood there is a connective tissue, and because of this connective tissue blood is made up of. Blood is basically alkaline. It has a pH about 7.4. It is salty also, and it is a viscous fluid. And main important thing is that blood is heavier than the water. Now see the board. So this blood is divided into two components. I have already told that first is the plasma and second is the blood cells. Plasma is about 55% and blood cells is about 44%. Here you can see these blood cells again divided into their cells. First is the Rbc, second is the Wbc and third is the platelets. And again this Wbc get divided into granulocytes and agranulocytes. And again these granulocytes get divided into neutrophils, basophils and eosinophils. And these agranulocytes get divided into lymphocytes and monocytes. But first of all we will see the plasma, and after that we will go for a blood cells. We already know there is a 55% plasma is present in the blood. So this plasma has a off-white color, that means somehow creamish color, as well as it is a slightly alkaline, and of course it is a viscous fluid. And plasma consists the water, and water ratio will be 90%, as well it also consists proteins. It will only 7 to 8%, as well inorganic salt also present in the plasma. It is about 1%, and some other components also present in the plasma, and that will be also 1 or 2%. So that's all for the plasma. Now we will see the blood cells. We have already seen that blood cells get divided into three types: Rbc, Wbc platelets. Basically, these blood cells also called as a form cells or a form elements. Why it is called as a form elements? Because these all blood cells, that is RBC, WBC and platelets, togetherly called as a form elements. And further, this human blood get divided into three elements: RBC, WBC and platelets. RBC means red blood cells or a red blood corpuscles. WBC is the white blood cells or a white blood corpuscles. And next is the platelets. First, we will see the RBC. This RBC is also called as a erythrocytes. And these RBC or erythrocytes are very more in number in our human body. This RBC has a somehow circular structure and it is a enucleated. Have you ever think that why blood has a red color? Because of the hemoglobin. And red blood cells, that means these cells have a red color. That means in the blood there is a hemoglobin is present and which carries the oxygen, and that's why this RBC has a red color and this cells has a red color. Sometimes doctor said that you have to check your RBC count or a WBC count or a platelets count. So for every cell, that means for RBC, for WBC or for platelets, there is a particular total count is decided. Total count of RBC, total count of WBC, total count of platelets, and on the basis of this RBC count, range is decided. And in male and female there is a different average number. In male there is a 5.1 to 5.8 million per mm cube, and in female average number of RBCs 4.3 to 5.2 million per mm cube, and every cell has their lifespan. So that's why this RBC has a 120 days of a lifespan. And when this RBC get formed, and that process is called as a erythropoiesis. That means in the erythropoiesis, RBC get formed, and that particular process is called as a erythropoiesis. Basically, these RBC are produced from the hematoblast or reticulocytes. That means when RBC are forming, at that time there is a particular location in adults and in a fetus. So in fetus, RBC form in the liver and spleen, whereas in adults RBC get formed in the bone marrow. It is also called as a red bone marrow. Sometimes students, you have seen that or you have heard that when a person causes a cancer, at that time doctor said that you should do bone marrow. Why it is happening, especially in the blood cancer? Because of bone marrow. In the adult, bone marrow is the main organ for the forming of a RBC, that means a red blood cells. and in the bone marrow this RBC get formed. That's why for the formation of a RBC there is a vitamin B12, folic acid, heme protein are essential. When the RBC lifespan get ended, that means when 120 days over, at that time this RBC get destroyed. But where? In the spleen and the liver. When this RBC count get increased, at that time it is called as a polycythemia, and when this RBC count get decreased, at that time it is called as a erythrocytopenia. And this RBC also contain the enzyme carbonic anhydrase. We have seen carbonic anhydrase. It is an enzyme in a previous lecture. So, main function of this RBC are basically it is a helpful in a oxygen and carbon dioxide which is transported in the respiration. Along with it also maintains the pH of a blood, also viscosity of the blood. And blood clotting also happens because of the RBC. RBC is mainly responsible for the blood clotting. And we already seen that there is average number different in male and female. So there will be blood ratio will be different in male and female, and this ratio is called as a hematocrit. And this hematocrit is the ratio of the volume of RBC to total blood volume of a blood. On the basis this hematocrit is identified. Now we will see the WBCs. WBC is the white blood cells, or it is also called as a leukocytes. Basically, white blood cells means cells which don't have any color. That means these leukocytes are colorless. It has a nucleus and also it has a amoeboid cells. Why it is called as a amoeboid cells? Because its movement is similar as a amoeba. It can move out of the capillary walls, and this all process is called as a diapedesis. So it called as a amoeboid cells, and these WBCs are larger than the RBCs. I have already told that RBC, WBC or platelets, every cell has their average number, and on the basis of this average number, cells range is decided. So in the WBC, there is average number is 5000 to 11,000 per mm cube of the blood. When the WBCs get decreased, at that time there will be cause of a leukopenia. Leukopenia means when patient have a HIV, AIDS or a TB, or they exposed to any radiations, or patient faces some shock, or patient exposed to any radiation, at that time WBC get decreased and they will cause a leukopenia. But when temporarily WBC cells will increase, at that time patient will cause a leukocytosis. It is caused only by a infection because it is a temporary increases. That's why. Or it is also increased during pregnancy or newborn babies, because it is a temporary change. So they will cause a leukocytosis. But when these WBCs get increases uncontrollably, that means uncontrolled increase of WBCs only happen in a blood cancer, and it is called as a leukemia. Leukemia, it is a one type of a blood cancer, and in the blood cancer WBC gets infinitely increasing number. They continuously increase their number and multiplies their numbers, and at this stage it is called as a uncontrolled growth of a WBCs or uncontrolled count of a WBCs. At that time leukemia causes, that means a blood cancer. That's why this WBC is called as a defense mechanism. Why it is called as a defense mechanism? Because of this WBC has a main function, and that is a protection. And because of these WBCs in our body, we can fight with any infection, we can face any infection, or because of the WBC our immune system gets stronger or weaker, only on the basis of the number of or a range of or amount of that WBCs. We have already seen that WBC is a colorless as well as it is a irregular. I have already told that it have a amoeboid movement. Like that, amoeba don't have any particular structure, same as like that this WBC has an irregular shape and structure. This WBC don't have a particular structure, and it is a nucleated. That means in WBCs there is a nucleus is present, and this WBC is divided into two types, granulocytes and agranulocytes. Basic difference is granulocytes means in the granulocytes there are granules are present, and in agranulocytes there are granules are not present. First we will see the granulocytes and their types. So these granulocytes are divided into neutrophils, basophils and eosinophils. These granulocytes have a granular cytoplasm, and that's why it is called as a polymorphonuclear leukocytes or a PML. And in these granulocytes they have a three types, and in every three types the nucleus has a different shape. You can see this is the neutrophils, this is the basophils and this is the eosinophils, and this has a different structure of a nucleus. It is also has a different structure of a nucleus, and it is also different structure of a nucleus. And these granulocytes are made up of stem cells, and stem cells also various types. So there is a one type of stem cells called as a myeloid, and these granulocytes is made up of a myeloid stem cells. But when these granulocytes get formed, at that time further it didn't divide, it didn't differentiate. And in the granulocytes and in the WBC there are 72% granulocytes are present, and remaining 28% are agranulocytes. So we have seen that granulocytes have a granules. So main function of these granules are they secretes the vesicles as well as they secretes the enzymes. granules has a staining property. That means these granules stain ourselves by various stains, and on the basis of these stains, these granules are divided into three types. First is the neutrophils, second is the basophils, and third is the eosinophils. First is the neutrophils. You can see this is the diagram of a neutrophils. You can see here is the granules also and nucleus also. First of all, I have told you each and every type nucleus will have a different structure and shape. You can see here the nucleus has a particular structure, and in that there is a granules are present. So these granules are very fine as well as very large in number. And I have already told that on the basis of the staining property, these granules are distributed. So these neutrophils get stained by only natural stains, that is a dyes. And these neutrophils are about 70% which is present in the WBC. And these neutrophils are able to move like a amoeba. It has a amoeboid movements. And also it has a phagocytosis property. What do you mean by phagocytosis? That means the pathogens which enters in our body, these pathogens get destroyed by the phagocytosis method. So it is called as a phagocytosis. And one more thing, in neutrophils there is a pus mixture. What do you mean by pus mixture? So pus mixture is the mixture which consists damaged tissue, dead microbes and dead neutrophils. So this mixture is called as a pus mixture. Second is the basophils. You can see here also these basophils has a different structure of a nucleus, and you can see granules also present, but these granules are a large in size as compared to neutrophils, and it is a very few in a number, and it is stained by a basic stain, and basic stain is the methylene blue. So that's why these granules will stain by a blue color. When you will observe in the microscope, at that time this granule will have a blue color, then it will be a basophils. These basophils also called as a cyanophils. But this basophils is a non-phagocytic. That means it didn't destroy the pathogens. That's why it is about 0.5 to 1% present in the WBC. It don't have a property like phagocytosis. It is a non-phagocytic. So that's why it is present in the infected area or any allergic condition. And main thing is that these basophils secretes the heparin, histamine, serotonin, etc. Next is the eosinophils, and this eosinophils is also called as a acidophils. That means this will stain by a acidic stain, and acidic stain is a eosin, and that's why it is called as a eosinophils, and it will get a red color. That means this granule will get a red color. When you will observe in a microscope, at that time when granules observed in a red color, at that time it will be a eosinophils. And you can see here in this diagram, this nucleus also has a different structure and a shape. So these eosinophils are about 1 to 3% present in the WBC, and it is have a phagocytosis property. So it will destroy the pathogens antigen antibody complex also. And this number of eosinophils get increased in allergic conditions, and that's why it will show the antihistaminic property. Antihistaminic property, that means it will increase their numbers in allergic condition for the fighting the or destroying the pathogens. And it is also produce the antitoxins. That's why it is responsible for the detoxification. When they produce antitoxins, at that time these antitoxins will helpful in a detoxification. So this is all about granulocytes. Now we will see the agranulocytes. I have already told that 72% is present in a granulocytes and remaining 28% are agranulocytes. So these agranulocytes get divided into lymphocytes and monocytes. Basically these agranulocytes are made up of stem cells, that is a lymphoid stem cells, and again it get divided by the mitosis process. But as compared to granulocytes, these agranulocytes have a large nucleus. You can see in the diagram, here is the nucleus are large as compared to granulocytes. So these granulocytes get divided into two types. First is the lymphocytes and second is the monocytes. So first is the lymphocytes. Actually it is a very small and it has a spherical nucleus, you can see. And main thing, in agranulocytes there will be a granulocytes will absent. So you can see in both diagram, that means in both lymphocyte and monocytes, there will be no granules are present. It will be absent. So these lymphocytes will be about 25 to 30% in the WBC, and this lymphocyte will have two types. First is the B lymphocyte and second is the T lymphocyte. B for a bone marrow and T for a thymus. That's why in B lymphocytes bone marrow included, and it is helpful in antibody production or our production of our immune system. That's why bone marrow is a very important part. Next is the T lymphocyte. I have already told that T for a thymus. So these T lymphocytes will present in the thymus and it will helpful in cell mediated immune system. That means it will helpful in T cells, killer T cells, memory T cells and suppressor T cells. That means this killer, memory, suppressor, all these are function of a T lymphocytes. And this is also types of T lymphocytes. And main function of the lymphocytes are to maintain our immune system, as well as it is responsible for antigen antibody formation. Next will be a monocytes. You can see there is a granules are absent, but this nucleus is a large inner size and it has a bean shape or a kidney shape. This monocytes is about 3 to 5% present in the WBC, but these monocytes are very actively motile. It has a phagocytosis property, so that it destroys the pathogens, bacteria, damaged tissue. These all destroyed by this phagocytosis property. Now we will see the platelets. This platelet is also called as a thrombocytes. So these platelets have a fragmented structure. We have first seen the WBC and RBC, which has a circular structure or a spherical structure, but these platelets have a fragmented structure, and it is get formed by the large cell. That means large cell get fragmented into small cells, and this is called as a megakaryocytes. And these platelets or thrombocytes are produced in the bone marrow. So this fragment will be very small in number and very small in size, as well as it has a somehow oval shape. And main thing is that platelets don't have a nucleus. As I told you, this RBC, WBC, also platelets have a particular average number. On the basis of that, range is decided. So these platelets have an average number about 2.5 to 4.5 lakh per mm cube of the blood. That means these platelets will present in the blood 2.5 to 4.5 lakh per mm cube. When these thrombocytes or platelets decrease their number, at that time human will cause a thrombocytopenia. And thrombocytopenia means a blood hemorrhage. That means the internal bleeding. When internal bleeding occurs, at that time thrombocytopenia get caused. And when blood hemorrhage occurs, at that time this platelet secretes the platelet factors, and these platelet factors are very important in the blood clotting. And when the blood clotting didn't occur, at that time internal bleeding get started and blood hemorrhage get started, and in a very large number this blood get lost. But this can be get coated or sealed with a ruptured blood cells or a ruptured blood vessels by forming a particular platelet plug or a thrombus. because this thrombus or a platelet plug secretes the serotonin, and this serotonin is a vasoconstrictor, and because of this vasoconstrictor internal bleeding will stop, and also because of this platelet plug or a thrombus bleeding will also get stopped. Now you can see here is a one diagram called as a blood smear. When we will take a blood smear and when we will observe it in the microscope, at that time we will see RBC, WBC, platelets. So when there will be a red dot or a red color cells, this will be a RBC. When there will be a bluish color or a violet color, at that time there will be a WBCs. And when there will be very fine, very small dot-like structure or a granule-like structure, it will be a platelets. And when we will observe that WBC and types of WBC, at that time, if you have to see neutrophils, at that time it will stain by a neutral stain. Second, basophils will stain by a methane blue, that means blue color, and eosinophils will stain by red color, that is a acidic stain. So today we have seen the circulatory system in a human, and it includes the blood vascular system, and in that blood vascular system we have seen the composition of a blood. Also we have seen the lymphatic system, and it consists the plasma and blood cells. We have seen the composition of a plasma, as well we have seen the blood cells also, and these blood cells get divided into RBC, WBC and platelets. Further, WBC get divided into granulocytes and agranulocytes. Granulocytes get further divided into neutrophils, basophils and eosinophils. And next, agranulocytes again divided into two types, lymphocytes and monocytes. And further, we have seen the platelets, and also we have just observed the blood smear, and in blood smear how cells are observed, that we have seen. So that's all for today. In next lecture we will see heart and the external structure and internal structure and mechanism of the heart. Thank you.